This lecture covered Chapter 8 and has the title How to Obtain a Clone of a Specific Gene. The previous chapters dealt with how we can clone or study genes that we have nucleotide sequence for. This chapter offers an overview of the toolbox available to us in order to identify the sequence of a particular unknown gene. How do we find a gene? Well, this depends on what kind of protein that the gene encodes. If the gene is an enzyme, or if the gene encodes an enzyme, we can use the enzyme activity in order to identify the gene of interest. We can use complementation, which is an in vivo technique, in order to clone resistance genes and metabolic genes. We can use Zymography to detect enzymatic reactions in vitro, provided that we have some way to detect this reaction. We can use nucleic acid sequence similarity, technologies like Southern Blot, in order to identify similar genes. And we can use protein structure using technologies like Western Blot. A common first step, regardless of the technique used, is to create a genetic library if it is at all possible. Libraries are populations of cells, usually E. coli, each carrying a different piece of DNA from a particular source organism. There are two kinds of libraries, two kinds of DNA libraries, genomic and cDNA libraries. Construction of a genomic library starts with the fragmentation of genomic DNA from a particular source. This fragmentation can be mechanical, or it can be a partial digestion with restriction enzymes or a mixture of enzymes. Mechanical fragmentation has the advantage of being random, but the DNA is damaged in the process, so it has to be prepared or has to be processed for the subsequent cloning step. Restriction enzymes doesn't have this disadvantage, but on the other hand, we have to make sure that not all restriction sites are digested, because then the representation of certain sequences will be low in our library. The fragments, the linear DNA fragments, are then cloned in a plasmid vector, and these resulting recombinant DNA molecules are transformed into bacteria, usually E. coli, in such a way that each E. coli cell has only one insert, one type of insert. cDNA libraries are different in that what we clone into the recombinant vectors is complementary DNA of messenger RNA. These complementary DNA molecules are produced by reverse transcription, and in this case we don't have the fragmentation step, and in theory each clone in the library represent an expressed gene in the source organism. Usually in both of these techniques there is some kind of size selection for the inserts, because you want, usually depending on the capacity of the plasmid or other reasons, you want a certain size range in your library. cDNA synthesis from eukaryotic sources always starts with the synthesis of a first DNA strand, this one here, using reverse transcriptase and a poly-T primer. The poly-T primer will bind to the poly-A tail of eukaryotic mRNA, and this will provide a starting point for the reverse transcriptase. There are many variations of this technique, and they mostly differ in how the second DNA strand is synthesized. In this case, the original RNA strand is fragmented by adding RNase H, and these fragments will still sit on the DNA copy of on the cDNA on the first strand, and these fragments will act as some kind of primers, RNA primers, to synthesize the second DNA strand using a DNA polymerase and dNTPs. This means that the resulting molecule is probably not the complete gene, because it's not very likely that there is one of these RNA fragments exactly at the beginning of the gene. So the resulting library in this case will not have many complete genes. But if this is not an important point in this case, then this is a viable option in order to create a cDNA library. There are some variations of the technique in order to increase the representation of complete genes in the library. And we will discuss these later. Important differences between genomic and cDNA libraries are summarized in this table. cDNA libraries usually only contain the coding sequence, or the coding part of the genome, so it's usually smaller. The technique that I showed in the previous slide implies using eukaryotic mRNA, as prokaryotic mRNA have no poly-A tail, so creating the first strand is more difficult. Prokaryotic mRNA is also a much less stable molecule than eukaryotic mRNA. However, the reason for making a cDNA libraries as opposed to a genomic libraries in the first place is the presence of introns in eukaryotic genes. These are not usually present in prokaryotic organisms, so there is perhaps little reason to make cDNA libraries of prokaryotic organisms anyway. Coverage and representation are important properties of a DNA library, and we can use this equation in order to calculate the number of clones that are required in our library, and this number depends on the probability that we require for the presence of our gene and the average size of each DNA fragment in our vectors and the size of the genome. So we can see we need more clones if we want a higher probability to find our gene, or the genome is larger. On the other hand, if each plasmid carries a larger insert, we need less clones. For instance, if we want to find a sequence in Saccharomyces cerevisiae, and we have a library of 2,000 base pairs, average size, the Saccharomyces cerevisiae has 12 million base pairs, roughly. This means that, according to this equation, we need a library of roughly 28,000 clones in order to find our gene. Of course, each insert of the library has to be large enough in order to contain the gene we are interested in. The direct selection of antibiotic resistance genes is, of course, simple and a very efficient method, but antibiotic resistance genes are a very small fraction of all the interesting genes we want to clone. Complementation, on the other hand, is a technique that's very similar, but it can be used to clone a much wider array, a much wider range of genes. In this example, we show the cloning of the TRPA gene using a mutant E. coli that has a mutation for that same gene. This TRPA gene is a metabolic gene in the biosynthesis of tryptophan, and this mutant organism cannot grow without tryptophan in the medium, so it's one of many possible mutants in that metabolic pathway. The rest of the procedure is similar. We make a DNA library. Only the one gene that or the one plasmid containing the TRPA gene will make it possible for the library host organism to survive on a medium without tryptophan. So this is a technology very useful for metabolic genes that have a very clear phenotype. In this case, this is growth or not growth. But one problem or one complication is that we need a mutant in order to perform this procedure. And this may be counterintuitive, but in order to clone the TRPA gene, we need a mutant for that very same gene. But we don't actually need to know the gene of a particular, the gene sequence of a particular gene in order to create a mutant for that gene. So how can we then create auxotrophic mutants, such as the TRPA mutant in the previous slide, without actually knowing the DNA sequence of the gene? This slide outlines the strategy for generating auxotrophic mutants in the uracil metabolic pathway, which is similar to the one you would use for the TRP metabolic pathway. The first step is to subject a population of cells to some kind of mutagenic treatment. It can be UV light, for instance. Here in this population, all kinds of random mutants will arise. Most of the cells will not have any mutations at all, but random mutants will arise, and some of these mutants will be affected in the metabolic pathway for the production of uracil. There can be several different genes that can be affected, and in this case they're symbolized by URA1 and URA2. So these are two different genes, but they are both affected in the metabolism of uracil. Then we take this population of cells and cultivate it in a medium without uracil. This means that all mutants that are not affected, all of these that are not affected in the URA genes, will start to grow, whereas these mutants affected in the uracil pathway will stay in stationary phase or not grow. Then we add nystatin, or some kind of similar antibiotic. This is the molecule, nystatin. It's produced by penicillin mold. And this antibiotic has a particularly interesting property. It kills cells that are actively growing. It selectively kills the growing cells. This means that all of these mutants that are not affected in the URA1 or URA2 pathway, URA1 or URA2 genes here, will be killed. And if we then plate these remaining cells on a medium with uracil, but without nystatin, of course, then these clones that survive here will be at least enriched for genes in the URA pathway. But how do we separate the population of mutants in the different genes? We assume that we now have a populations of of mutants in the uracil pathway. This is the uracil pathway right here. And we have several genes in this pathway, and mutations in either of these will cause the phenotype that we observe in the URA1 and URA2, or the population of the URA mutants. We need some way of separating them so that we can know which one to use for cloning a specific gene. So this is the reason why we might want to use Saccharomyces cerevisiae instead of E. coli in this case. Well, why Saccharomyces cerevisiae instead of E. coli in this case? Well, it depends on the sexual cycle of Saccharomyces cerevisiae. It has 2 stages to life cycles here. We have a stage of haploids. This means that there is only one copy of each gene, and there are 2 types or 2 sexes, A and alpha. These 2 can combine together and mate, and then create diploid cells that can grow happily, just as happily as the haploids. And these diploids can then be sporulated again in order to regain the haploids. So this cycle can be used in order to classify the metabolic mutants that we have in our population. So how can we use this sexual system in order to classify our mutants? We imagine now that we have a population of URA mutants in the A type of cells, these green ones, and we have also a population of random mutants in the alpha type, these pink ones here. Imagine that we have 5 mutants of each kind. Usually we would have more, but we simply mate all of them in every possible combination. So we would need to combine 25 different pairs of mutants. If we imagine that we mate 2 cells that are affected in the same gene, this means that the URA1 gene is, in this case, inactive. This URA1 gene will be inactive also in the diploid cell, this big one here. So the same phenotype will be observed in the diploid. If, on the other hand, the cells are affected in different genes, here URA1 and URA2 in the alpha cell, the resulting diploid will have no phenotype at all, because the URA1, the active one in the alpha cell, will complement the inactive one in the A cell, and vice versa for the URA2. So here we will have a cell that grows without uracil in the medium. If we do this enough times, we can classify all the mutants in something called complementation groups. So how do we perform this complementation in a practical sense? We first start with 2 non-selective plates where we streak our alpha mutants in lines like this, and another one where we streak the A mutants in a line like this. We have now 4 mutants of each kind in this example. Then we transfer these mutants to a third plate using a piece of cloth like that, so that we obtain a cross pattern. This plate is also non-selective, but we have the A mutants here on the x-axis and the alpha mutants, the A mutants here, and we call them A, B, C, and D, and 1, 2, 3, and 4. Then we use another piece of cloth to transfer this cross pattern of cells onto a selective plate, and we can observe in this case that some combinations of cells here. In each cross there is a certain combination, and these haploid cells will form diploids. They will fuse and form diploid cells. Only some of them will grow, meaning that they are affected in different genes. And what we say here is that mutants C and D and 1, C, D and 1, for example, constitute a complementation group, because these mutants do not complement each other, meaning that they are affected in the same gene. Most metabolic genes were actually defined in this way long before there were any molecular tools, or before we even knew how to manipulate DNA in vitro. This is why many metabolic genes have quite strange names. In Saccharomyces cerevisiae, for instance, there are 10 URA genes, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. And these were named before we knew what kind of enzyme they encode. There are many examples of this other than uracil. All the other important compounds within the cell, for which there is a very strong phenotype if the metabolic capacity to produce them are knocked out. Some of these auxotrophic genes we use for as selection markers in yeast in order to maintain plasmids and other genetic constructs. Complementation is a powerful method, but it is less useful for genes with a weak or no phenotype at all when deleted. Zymography is an alternative technique that can be used to identify enzymes for which there is a reaction that can be detected visually by a change in color, like we can see here. It's especially useful for genes encoding secreted enzymes. The procedure starts with separation of proteins on a standard SDS page gel. The SDS detergent is very aggressive and it denatures the proteins. After separation, the SDS is washed away and replaced by Triton, which is also a detergent but a less aggressive one. This leads to at least partial renaturation and refold of the proteins in the gel. Then the gel is developed by adding the appropriate substrate and other chemicals in order to visualize the reaction of the enzyme. For instance, in this gel here, protease mixtures were separated, and on a gel that contains gelatin and Comasi brilliant blue. So the Comasi brilliant blue colors the gelatin in the gel. Here, where you see the white spots, we have a protease that digested the gelatin, and hence we see no coloring of the gel. Zymography can be extended to reactions or detecting enzymes that catalyze reactions that consume or produce NADH or NADPH, or consume or produce ATP. In this example here, I show the detection of lactide dehydrogenase. Normally this enzyme catalyzes the production of lactic acid from pyruvic acid. In this case, we can detect the opposite reaction. We have production of pyruvic acid and the production of NADH. And this NADH is used as a substrate for an enzyme called diaphorase, which can catalyze the reduction of a dye called tetrazolium to another dye called formazan. And this change in color from yellow to red we can detect. This means that we can separate also other kinds of enzymes on a gel in the same way as I showed in the previous slide, and we can detect many kinds of metabolic enzymes. Genes have neither mutant phenotype or activity, such as structural genes, for example. These can still be detected if we have antibodies for the protein or a similar protein. We can use a technique called Western blot to first separate the proteins of interest on an STS page gel, the same kind of gel as in the previous slide. We use electricity to blot over the proteins on a membrane, and here we can detect these proteins on the membrane with an antibody. This antibody is usually detected with a second antibody, which is labeled with an enzyme that has an easily detectable reaction or even emission of light. This use of a secondary antibody means that you don't have to label your primary antibody, which is an advantage because you want to switch this antibody depending on the protein you want to detect. An important problem to solve is, of course, how to clone a gene for an enzyme if the only thing we have is a purified protein. The first step in this process is to simply cut out the protein from the gel or the blot and isolate and purify the protein. We can then use mass spectrometry in some cases, or we can simply sequence the protein by a technology called Edman degradation. This is a quite simple technique where a molecule called PITC is linked to the N-terminal of the protein. Then we can split the protein between the first and the second amino acid here. This PITC is the red triangle here. And we can separate and analyze by HPLC. We can then repeat this process, and we can in this way sequence the protein sequence at least 5 or 10 amino acids. We can use the protein sequence information discussed in the previous slide to guess the DNA sequence that should be present in the gene encoding for the protein. This is of course complicated by the fact that translation is degenerate and several DNA sequences can encode the same protein sequence. If we have amino acid sequence information like the one we have here in the picture, if possible, we should choose stretches of amino acids for which there is only 2 codons. There are up to 4 codons for some amino acids. In this case, we chose the trip, asp, glu, asn, met sequence here. And this sequence of amino acids have only 2 codons each. This means that it is encoded by a DNA sequence of 18 nucleotides, and since there is 2 codons of each, we have 64 possible different nucleic acids that encode for this. This DNA sequence here can be synthesized as a mixture of oligonucleotides and used to find the gene that we are interested in. To do this, we use a technique called Southern blot, which is quite similar to the Western blot I just shown. DNA fragments are separated on a gel. This DNA is transferred to a membrane, and the membrane is mixed with a labeled probe DNA and washed and then developed with an X-ray film. And we can detect where we have specific similarities between the DNA in the gel and the probe. In this case, this is a general technique that can be used in many other cases. In this case, we would use the sequence information in the previous slide, synthesize the 64 different labeled DNA molecules and probe, and then discover which DNA fragment contain a sequence that is similar to any of the ones that we synthesized. DNA is commonly labeled radioactively by one out of 4 common methods. The first 3 methods are shown here, A, B, and C. They all rely on a mixture of DNA polymerase and radioactively labeled nucleotides. Single-stranded regions of DNA, in this case, in method A, can be repaired by radioactive nucleotides and the DNA polymerase, or single-stranded ends of the molecule, generated in this case by a restriction enzyme, can be filled in by the same method. Or alternatively, the probe DNA, the original probe DNA, is denatured and random hexanucleotides are not ligated but annealed to the DNA sequence, and radioactive pieces of DNA are filled in by the DNA polymerase. In this case, we can say that the activity, that is, the amount of radioactivity per molecule, is higher in the C case here because we add more radioactive DNA effectively. In this case, we only add small stretches at the end of the molecule, so the activity of the probe is lower. DNA can also be labeled by adding a radioactive phosphate molecule to the end of the DNA sequence using T4 polynucleotide kinase. Here we use radioactive ATP, this molecule here, and a molecule that has OH hydroxyl groups on the 5' end, and by the reaction of the kinase, the ATP is turned into ADP, and we have radioactive phosphate on the 5' end. Even if our molecule already has 5' phosphate, we can still use the method if we add an excess of ADP in the mixture, along with the radioactive ATP, because the molecule, the polynucleotide kinase can transfer not only phosphate from ATP to DNA, but also DNA to ADP. So in this case, the end result will be radioactive DNA and a non-radioactive ATP in the end. So in essence, this phosphate here is transferred to ADP and replaced by this radioactive phosphate here. Radioactive labeling requires expensive equipment and safety precautions. So non-radioactive alternatives are becoming more popular. 2 examples are shown here. DNA can be labeled with a molecule called biotin, which is a small molecule, and it can be detected by some kind of other molecule linked to avidin. Avidin and biotin have very strong and specific affinity for each other. This molecule here can contain a fluorescent molecule, for instance, and the signal from the DNA can be detected by light. Alternatively, the DNA can be labeled directly with an enzyme. In this example here, horseradish peroxidase is glued to DNA by a molecule called glutaraldehyde, which acts as a kind of glue. This means that the specific DNA here of interest is labeled with this enzyme, and this enzyme is easily detectable by a reaction that produces light. Using luminol, horseradish peroxidase will carry out this reaction, which creates an unstable product, and this unstable product will generate an end product and a photon of light. This is an example of luminol in action. A curiosity is that the luminol reaction is also used to find blood, even if it was cleaned away in crime scenes, in criminal investigations. Southern blot can also be used in combination with the DNA library. So instead of separating our DNA on a gel and then transferring to a membrane, we plate clones of the DNA library on a plate, and then we transfer this DNA to a membrane. And we use, for example, the 64 oligonucleotide probes from slide 20, labeled here radioactively in the end. And we probe the membrane for clones that contain the gene we are interested in. In order to make a stronger detection, we can use several different probes, ideally for different parts of the gene, in order to more safely select the correct clone. If we have cloned a gene in one organism, we can use this information in order to clone related genes from other organisms. In this example here, we used a cloned gene of cytochrome C from yeast in combination with a gene library from Neurospora in order to clone or identify a clone that contains the cytochrome C gene of Neurospora. We can also use the same technique to find members of gene families. We have a cDNA from wheat, and we have a library from the same organism, and we can identify genes, similar genes, but in the same organisms.